Cardiovascular Physiology, the mechanics of cardiac muscle contraction. Welcome to our deep dive into the remarkable physiology of the heart. Today, we explore cardiac muscle contraction, the essential mechanical process responsible for pumping blood throughout the body. This phenomenon, known as excitation-contraction coupling, bridges the electrical signals within the heart to the actual physical shortening of its muscle fibers, a process that is nothing short of extraordinary. The foundation, sliding filament theory enhanced by calcium. At the core of cardiac muscle contraction lies the sliding filament theory. Here, actin and myosin filaments slide past one another, generating force. Cardiac muscle, however, introduces unique characteristics that set it apart from other muscles. The most crucial distinctions include the origin and pivotal function of calcium ions and the mechanisms that ensure the heart's contractions are graded and do not result in sustained, tetanic contractions. The architecture of cardiac muscle. Cardiac myocytes, though striated like skeletal muscle cells, exhibit several important differences. These cells are branched, typically possess a single nucleus, and form an interconnected network known as a functional syncytium. This network is made possible by specialized gap junctions, facilitating rapid communication between cells. The fundamental unit of contraction is the sarcomere, which contains thin filaments composed of actin, troponin, and tropomyosin, and thick filaments made of myosin. The sarcoplasmic reticulum serves as an internal reservoir for calcium ions, although it is not as extensive as in skeletal muscle. The transverse tubules, which are notably larger and more elaborate than those found in skeletal muscle, penetrate at the Z-lines and house a high concentration of L-type calcium channels. Importantly, cardiac muscle cells feature structures called dyads, where a transverse tubule is closely associated with the terminal cisterna of the sarcoplasmic reticulum. This arrangement differs from the triads observed in skeletal muscle. From electrical signal to muscle contraction, the sequence unveiled. Let us walk through the process that converts an electrical action potential into a mechanical contraction. The sequence begins with the propagation of an action potential through the heart's conduction system, which travels down the transverse tubules. This electrical wave opens voltage-gated L-type calcium channels located on the membrane of the transverse tubules. Unlike skeletal muscle, where calcium is sourced strictly from internal stores, cardiac muscle relies on a small but vital influx of calcium ions from the extracellular fluid. This influx acts as a trigger, binding to ryanodine receptors situated on the membrane of the sarcoplasmic reticulum. The engagement of these receptors prompts them to open, releasing a substantial amount of calcium ions from the sarcoplasmic reticulum into the cell cytoplasm, a phenomenon often referred to as the calcium spark. With the sudden rise in intracellular calcium concentration, contraction is initiated. Calcium binds to troponin C on the thin filament, inducing a structural change that shifts tropomyosin away from the myosin binding sites on actin. This allows myosin heads to attach to actin, forming cross bridges. The power stroke follows, causing the filaments to slide and the sarcomere to shorten. This cycle continues as long as calcium and adenosine triphosphate are available. Relaxation. The heart's opportunity to refill. Equally vital to contraction is the process of relaxation, enabling the heart to refill with blood. Once the action potential concludes, the L-type calcium channels close, halting the influx of trigger calcium. Multiple mechanisms promptly work to reduce cytoplasmic calcium levels. The sarcoplasmic and endoplasmic reticulum calcium ADPase, known as SERCA, is the principal player, actively pumping calcium back into the sarcoplasmic reticulum to replenish stores for the next heartbeat. The activity of SERCA is enhanced by phospholambin when it is phosphorylated, such as during beta-adrenergic stimulation. Another significant mechanism involves the sodium-calcium exchanger located on the cell membrane, which utilizes the sodium gradient to extrude calcium from the cell. A minor contributor, the sarcolemmal calcium ADPase, also assists in pumping calcium ions out of the cell. As intracellular calcium levels fall, calcium dissociates from troponin C, allowing tropomyosin to return and block myosin binding sites on actin. The cross-bridge cycle ceases, and the muscle fiber relaxes, permitting the heart chamber to expand and refill. Distinctive features of cardiac muscle contraction. One of the most crucial aspects of cardiac physiology is the lengthy refractory period, which serves to prevent tetanic, or sustained, contractions. The cardiac action potential is characterized by a prolonged plateau phase, a result of the slow influx of calcium ions through L-type channels. This extended depolarization aligns with a long effective refractory period, ensuring that a new action potential cannot be initiated until the muscle has nearly completed its contraction. This safeguard is essential, as sustained contraction would halt the heart's ability to refill and could be fatal. Graded contraction and the Frank-Starling law. Unlike skeletal muscle, which can display frequency-dependent summation and tetanus, the force of cardiac contraction is graded. That means the strength of each heartbeat can be adjusted based on the body's needs. Two fundamental mechanisms regulate this. The Frank-Starling mechanism, the heart's intrinsic ability to pump what it receives. When increased venous return stretches the ventricular muscle fibers during diastole, the overlap between actin and myosin filaments is optimized, and the myocytes become more sensitive to calcium. The result is a more forceful contraction during the next beat, ejecting the extra volume received. Inotropic state. The contractile strength can also be altered independently of muscle fiber length. This is achieved by adjusting the amount of calcium released during the calcium-induced calcium release process. Positive inotropy, resulting from sympathetic stimulation via norepinephrine, increases cyclic adenosine monophosphate, leading to enhanced calcium influx and more rapid calcium reuptake by circa. The net effect is a stronger, faster contraction. Negative inotropy, on the other hand, may result from parasympathetic stimulation, certain drugs, acidosis, or hypoxia, all of which decrease contractility. Bringing it all together in summary, excitation begins with the propagation of the action potential down the transverse tubules. Depolarization opens the L-type calcium channels, allowing a critical influx of calcium ions from the extracellular fluid. This trigger calcium binds to ryanodine receptors on the sarcoplasmic reticulum, causing a substantial release in what is known as calcium-induced calcium release. Calcium then binds to troponin C, initiating the cross-bridge cycle and shortening the sarcomere. Relaxation follows as calcium is resequestered by circa and extruded by the sodium-calcium exchanger. Once calcium dissociates from troponin, the muscle relaxes, and the heart refills with blood. This beautifully orchestrated system guarantees that each heartbeat is both powerful and precisely regulated, ensuring the body receives the blood flow it requires for every moment. Thanks for tuning in. If you enjoy the content, please like, share and subscribe. It's the Everything Surgery Podcast. Dissect the extraordinary.